Hello, my dear students. Welcome you all to Sri Jayendra Rank Zone classes. I am your mentor and your friend for the subject biology, and I will be taking the zoology part, that is animal science. My dear students, in the previous videos, we have started about the fluid connective tissue, which is the false connective tissue. It means the matrix is not having fibers, the matrix is clean, okay? So which basically includes blood and lymph. We discuss different components of blood like RBCs, red blood corpuscles, erythrocytes, WBCs, white blood corpuscles, leukocytes, and platelets. Now, the most important function of platelets is blood clotting. So now we are going to discuss about blood clotting. So blood clotting, also known as hemostasis. Hemostasis. Hemostasis is blood clotting or stasis of blood. Okay, stasis of blood, flow of blood. Blood flows from cut, okay, or wound, but after some times it stops automatically. It is called clotting of blood. So clotting of blood is a physiological function in which the traumatic part or the part which is having any abrasion or cut, it comes with a clot formation and the oozing of blood, means oozing out of blood, stops. Stops. Understood? The normal bleeding time is 1.3 minutes. Okay. And the normal clotting time or the coagulation time is 2 to 8 minutes. The normal bleeding time is 1 to 3 minutes, and the normal clotting time is 2 to 8 minutes. Sometimes the clot are also formed in intact blood vessel, means intravascular coagulation. Okay. Intravascular coagulation. Understood? Now coming to next, these intravascular clots, they are of two types. One is the thrombus clot. Thrombus is also known as vegetation. Okay. It is a static clot which grows bigger and ultimately block the blood vessels. It is a static blood clot. Like for example, this is the blood vessel. What is vegetation? Vegetation is the accumulation of dead RBCs. WBCs, okay, along with debris, along with saturated fats, triglycerides. They all keep on depositing on the membrane. Okay, they deposited on the membrane, and now because of this deposition, what will happen? Because of this deposition or the formation of a platelet plug, it forms what? A clot. Or it may be that because of any reason the internal bleeding is present. For example, the artery, it gets leaked out, or the artery, it gets punctured over here. Okay, means it becomes discontinued like this. So the platelet plug, it will attach to the endothelial wall, because of chemotaxy, and it will make a clot. So this type of clot is known as what? A thrombus clot. Understood? Thrombus clot is static, which grows bigger and ultimately block the blood vessels. If this clot is formed in the coronary vessels, means the blood vessels which are supplying to the heart, then coronary thrombosis, which can cause heart attack, angina pectoris. Okay. So if this clot is formed in coronary vessels, means the blood vessels which are supplying blood to the heart, walls of the heart, then it may lead to the formation of a coagulant or a clot, which will lead to non-supply of blood to the walls of the heart, and blood, it needs to be constantly supplied to the walls of heart for its regular and rhythmic contraction and relaxation, because heart walls, they need to contract and relax very fast and continuous, and that is why if it is not being done, then what will happen? The clot, it will this clot, it will lead to decrease in the blood flow, which will cause the non-supply of blood and oxygen to the heart walls, and these walls, then they will get fatigued and they will not contract and relax, which will cause the cardiac arrest or a heart attack. If it is formed in brain, okay, then it is known as what? Cephalic thrombus, which causes paralysis. Okay. So in case of cephalothrombosis or cephalic thrombus, so what happens basically? A clot which is formed, that clot will create a pressure, undue pressure on a nerve, or it will stop the flow, stop the blood supply. So the part of the brain which is not having the blood supply will undergo degradation or necrosis and leads to paralysis. Is it clear? Understood? Yeah. Now, so the thrombus clot is a static clot which grows bigger and ultimately block the blood vessels. If this clot is formed in the coronary vessels, then it is also known as the coronary thrombosis, which can cause heart attack. If it is formed in brain, then it is called cephalic thrombosis, which causes paralysis. Understood? Now, a second type of clot is embolus clot. Embolus clot, thrombus and emboli, they actually have same origin, but embolus mean it get when this clot or the part of the clot, it detaches because of the blood flow. It detaches like this. It detaches and move with the blood. Okay, so then it becomes a embolus. Moving clots which flow with the blood and ultimately dissolve in blood, they are known as what? Emboli. Understood? More harmful due to their moving nature. As they are moving in nature, they can easily clot any or easily block any small blood vessel or a arteriole. They can completely block any arteriole. Understood? That is why they are more dangerous. Okay. So the types of clot: thrombus clot and embolus clot. Now coming to next. This is a scanning electron microscope picture to show how fibrin cross-link polymers are formed. These fibrin cross-link polymers, they arrest the cells and they will make the clot. Understood? Okay. Now coming to next, mechanism of blood clotting. Mechanism of blood clotting basically includes what? It was proposed by McFarlane and co-workers. According to this theory, there are three steps in blood clotting. The very first step is release of thromboplastin. The very first step is release of thromboplastin. Thromboplastin is released. This thromboplastin is basically released because of the traumatic or injured tissue, the tissue which is injured. It synthesizes a exothromboplastin or a prothromboplastin, the precursor of thromboplastin, and platelets synthesize endothromboplastin. Okay, so basically the injured tissue, they synthesize exothromboplastin, and the platelets, they synthesize endothromboplastin. Both of these exo and endothromboplastin, they react with different proteins which are present in the plasma, and in the presence of calcium, very important, calcium divalent Ca2 plus ions to form prothrombinase enzymes or thrombokinase. Prothrombinase enzyme or thrombokinase. So please remember this, that thromboplastin is released by the injured tissue as well as from the platelets. The injured tissues, they release exothromboplastin, and the platelets, they release endothromboplastin. The endothromboplastin, it combines with the exothromboplastin in the presence of Ca2 plus ions to form prothrombinase enzyme or thrombokinase. Understood? This thrombokinase is basically formed by exo and endothromboplastin. This enzyme inactive heparin, very important. This enzyme thrombokinase, you can see thrombokinase, okay, which will dissolve the thrombus, which will dissolve the, thrombokinase, which will basically inactive or dissolve the anticoagulant, okay. So heparin is an anticoagulant, okay, which basically checks the formation, whether the intravascular clot is forming or not. Okay, so this heparin is a natural anticoagulant. So thrombokinase, it basically removes all the fibers, especially it will, degrade the fibrin cross-link polymers, and it will act as a anti-heparin. Okay, this enzyme inactive heparin, it will help as a anti-heparin. Conversion of prothrombin into thrombin, as we have to make a clot. The very first is production of exo and endothromboplastin. Then they combine to form prothrombinase or a thrombokinase enzyme, which act as an anti-heparin. Okay, it will initiate the clot formation. Conversion of prothrombin into thrombin. Prothrombinase enzymes convert inactive prothrombin into active prothrombin, active thrombin, in the presence of calcium divalent cation. So the conversion of prothrombin into thrombin takes place in the presence of thrombokinase. Okay, thrombokinase or a prothrombinase enzyme, it will convert inactive prothrombin into active thrombin in the presence of calcium divalent cation. Conversion of fibrinogen into fibrin. Fibrinogen is soluble protein of plasma. Fibrinogen is a soluble protein in plasma. So thrombin protein polymerizes monomers of fibrinogen into insoluble fibrous protein, which is known as what? Fibrin. Okay. So conversion of fibrinogen into fibrin. Fibrinogen is a soluble protein of plasma. Thrombin, it converts fibrinogen, which is a soluble protein. It will polymerizes the monomers of fibrinogen into a insoluble form, which is known as what? A fibrous protein, fibrin. So fibrin is a fibrous protein while fibrinogen is a soluble protein. Understood? So basically what is happening? Thrombokinase is the initiator. It will convert prothrombin into thrombin. Thrombin one formed, it will convert fibrinogen into fibrin. Fibrin forms a network on the cut or the site of injury in which blood corpuscles are got trapped. Okay. This form clotting of blood. After clotting, a pale yellow liquid oozes out, which is called serum. Okay. Which is known as what? Serum or serum, in which antibodies are present. Okay. So after clotting, a pale yellow liquid is comes out, it oozes out, which is known as what? Serum, which contains what? Antibodies. Understood? So blood corpuscles is equal to plasma. Blood minus corpuscles mean if we remove the corpuscles part from the blood, it becomes plasma. And if we remove the fibrinogen and the large proteins of the plasma, then it will become serum. So the difference between blood plasma and blood serum is what? That blood plasma is blood minus corpuscles and blood serum is plasma minus fibrinogen and large proteins. Mean the clotting elements. If we remove the clotting elements from plasma, it will become serum. Okay. Blood plasma is faint yellow in color. Blood serum is pale yellow in color. Okay. Blood plasma is faint yellow and blood serum is pale yellow. Understood? Blood plasma, it is having fibrinogen. Blood serum is not having fibrinogen. Okay. So these are some common differences. Now coming to next. This is the clotting cascade or the clotting pathway. There are two modes of clotting pathway. One is the intrinsic pathway and the another one is the extrinsic pathway. Although both intrinsic and extrinsic pathway, they merge in a common pathway. Okay. This is the common pathway. Okay. So the clotting cascade, it basically happens in a two-way process, either by extrinsic or either by intrinsic pathway. Okay. So before coming to these extrinsic and intrinsic pathway, I would like to tell you about these clotting factors. There are 13 clotting factors in blood clotting which are involved in blood clotting. These factors are mainly produced in liver. They are mainly produced in liver. Vitamin K, which is required in the synthesis of these clotting factors. Vitamin K, which is required in the synthesis of all these clotting factors. Other natural anticoagulants are hirudin, which is found in leech, anopheline, which is found in female Anopheles mosquito. That is why it keeps on sucking the taking up the blood meal without the blood getting coagulated. Lamperidine is present in petromyzon or lamprey. Okay, anticoagulants. Coumarin, it is obtained from plants. Warfarin, obtained from plants. They are commercialized as anticoagulants. Understood? To collect blood in bottle in blood bank, artificial coagulants are used. Okay, whenever blood is collected for any type of diagnostic test, so the blood is collected and that blood needs to be preserved. Okay, it should not clot. So we add sodium citrate, sodium oxalate, ethylene diamine tetraacetic acid. Okay, ethylene diamine tetraacetic acid, it will make a chelate with the calcium of blood, and calcium is essentially important in all the steps of blood clotting. Okay, these chemicals act as a calcium binding units and remove Ca2 plus ions from the blood. Okay, now bullet or a gunshot, it is having a mixture of EDTA, ethylene diamine tetraacetic acid. This EDTA basically it makes a chelate with the calcium ions. Okay, and that is why the person, it he or she loses his or her life because of excessive amount of blood loss. Okay, so EDTA, it is a artificial anticoagulant or a synthetic anticoagulant. Understood? These are the clotting factors. Factor number one, fibrinogen. Okay, it is a plasma protein. It is a plasma protein, you know. Source of obtaining it, liver. Pathway, it is present in the common pathway. It converts fibrin insoluble web-like substance or clot. So fibrinogen is converted into fibrin and it is involved in the common pathway. Understood? Then prothrombin. It is the second factor. Factor number second, prothrombin. Okay. It is a plasma protein, liver. It is also involved in common pathway, converted to thrombin, converts fibrinogen into fibrin. You know that, okay? Prothrombin is converted into thrombin and thrombin converts fibrinogen into fibrin. Factor number three, tissue factor, which is a plasma membrane glycoprotein. Tissue factor is a plasma membrane glycoprotein. It is present or obtained from tissues. It activates extrinsic pathway. The most important, please remember. That factor number three, tissue factor, it basically, it initiates the extrinsic pathway, okay, and it is obtained from plasma membrane proteins, okay, glycoproteins, and the source is tissue cells. Factor number four, calcium ions. They are inorganic ions, source plasma, and they are needed for essentially all stages of coagulation process, always present. They are always present. Factor number five, proacylserine. It is a plasma protein. Present or source is liver and platelets, and it is involved in common pathway. So factor number one, factor number two, factor number five, they are involved in common pathway. So I'll write it down. Factor number one, factor number two, and factor number five. Calcium is factor number four, okay, which is involved in all the stages of coagulation. Okay. Factor number three initiates the extrinsic pathway. Factor number six is unknown. Unknown factor when we don't know the properties. Then factor number seven, it is proconvertin. Proconvertin, which is a plasma protein, liver, both extrinsic and intrinsic pathway. So it is present in both extrinsic and intrinsic pathway. But common pathway is something different. Common pathway is mean when extrinsic and intrinsic pathway they merge, okay, at a point. That is the common pathway. So proconvertin factor number seven is needed by both extrinsic and intrinsic pathways. Factor number 8, anti-hemophilic factor. It is a plasma protein, source liver, lung, capillaries. It is an extrinsic pathway. So factor number 8, intrinsic pathway. Factor number 8. Okay. Deficiency results in hemophilia A. If the factor 8 becomes deficient, then the person suffers from hemophilia type A. Factor number 9, known as plasma thromboplastin component PTC, plasma protein, liver. It is also an intrinsic pathway. Deficiency results in hemophilia B. Factor number 10, stored power factor. It is a plasma protein, source liver. It is a common pathway. So add factor number 10. Okay. Then factor number 11, plasma thromboplastin antecedent, PTC and PTA, both are very confusing. PTC is factor number 9 and PTA is factor number 11. Plasma protein, liver, intrinsic pathway. Sorry. 11. Intrinsic pathway. Okay. Then factor number 8, factor number 9, factor number 11, they all are in intrinsic pathway. 9, factor number 11, intrinsic pathway. Understood? Then factor number 12, Hegman factor. It is a plasma protein activated by negatively charged surfaces like glass. Okay. Liver, it is found in liver. Intrinsic pathway, factor number 12. It activates plasmin. Okay. It initiates the clotting in vitro when inside a living organism. It initiates the clotting in vitro. Okay. Activation initiates inflammation. So it is also a progressor of inflammation. All these are questions, right? Plasmin is secreted by factor number 12, which is the intrinsic pathway factor. Factor number 13, it is a fibrin stabilizing factor, plasma protein. It is present in liver and bone marrow. It cross-links the fibrin, forming a strong, stable clot. Okay, so it is an unknown that factor number 8, it belongs to which category. Okay. Okay. Now, factor number 6 is basically properties are not known, but it is believed that it behaves as the factor number 5. So common pathway factor number 1, 2, 5 and 10, sorry, factor number 1, 2, 5, 10. 1, 2, 5, 10. 1, 2, 5, 10. Common pathway. 8, 9, 11, 12. Intrinsic pathway. Okay. Factor number 3. Factor number 3, extrinsic pathway. Okay. Factor number 7, both extrinsic and intrinsic pathway. Understood? Now, these are the clotting factors. Now, I'll show you the clotting cascade. Now, what is exactly happening? See, the intrinsic pathway, it is activated, factor number 12. It is inactive. Firstly, it is inactive. Okay. So factor number 12, you can remember the name of factor number 12 from here. Factor number 12 is Hegman factor. Okay. The factor number 12, it gets activated to activated form. This is activated form, double tick. In the presence of calcium ions, activated form of factor number 12, it further activates the factor number 11. Activated factor number 11, it activates the factor number 9. Okay. In the presence of calcium ions. Activated 9, it converts 10 into activation. Now, the conversion of activation of factor number 10, it includes or it involves calcium ions. This PL is platelet phospholipids. Platelet phospholipids, PL is platelet phospholipids. And factor number 8 is also involved in the conversion of or activation of factor number 10. So please remember that activation of factor number 10 is very important because activation of factor number 10, it involves the calcium ions, the platelets, phospholipids, okay, and the factor number 8. Understood? Okay. That is why I'll tell you factor number 7, that is why it is written that factor number 7, it involves in both extrinsic and intrinsic pathway. Because this factor number 7, it is involved, it is a tissue factor, which involves in the conversion of factor number 8 as well. Okay. So now when 10 is activated, this 10 in the presence of factor number 5, factor platelets, phospholipid and calcium ions, it converts prothrombin into thrombin. So this is the common pathway. This is the common pathway. Okay. Prothrombin be converted into thrombin. Thrombin then convert fibrinogen into fibrin. Fibrin is polymerized to form fibrin clot. And then factor number 13, which stabilizes the fibrin clot, and clot is confirmed. Okay. So need not to actually go through each and every step. Just remember that which factor is involved in which pathway. Okay. That is the question. And what are the different factors involved in common pathways? Now, blood groups. Antigens of blood groups, they are present on the surface of RBCs, also called agglutinogens. Okay. Antibody or blood group antigen is present in the serum or the plasma, which is called agglutinin. So antigen of blood groups, they are present on the surface of RBC known as agglutinogen. And antibody of blood group, it is present in the serum or the plasma, which is known as what? Agglutinin. Understood? Antigen and antibody, they are special type of glycoproteins. Both antigen and antibody, they are special type of glycoproteins. Blood groups, there are four types: blood group A, B, AB, and O. A, B, O, they are discovered by Landsteiner. Okay. AB discovered by D. Castillo and Strolai. The blood group AB was discovered by D. Castillo and Strolai. ABO, they were discovered by Landsteiner. Blood groups O is universal donor and blood group AB is universal acceptor. Blood groups, they are the example of multiple alleles, polymorphism, DNA polymorphism. Okay, polymorphism means they basically show that a single trait is carried by different phenotypic characters. Okay, so this is a polymorphism. For gene of blood group, three alternatives are present. Gene of A and B are considered as dominant alleles. Dominant alleles, okay, they are represented as IA and IB, where I represents agglutinogens. Okay, so IA and IB for AB blood group. Okay, IA and IA. This is also a A blood group in which this capital A is dominant form and small a is recessive form. Okay, then IB, ID, heterozygous. This is heterozygous. Heterozygous. This is also heterozygous. Understood? Homozygous, it is written like this. I'll tell you in the next slide. So A and B are dominant over blood group O. They give their expression in homozygous as well as heterozygous conditions. So blood group A and B are due to dominant genes, allele A and B. Understood? So if a person is having a blood group AB, it simply means it is an example of codominance. AB blood group, it is an example of codominance. The antigen is A, then antigen is A, antibody is B or anti-B. Okay. It received blood group A and O, donate blood group A and AB. Donate two blood group A and AB. Blood group is B, then antigen is B, and antibodies against A. So it can receive from B and O and it can donate to B and AB. Okay. Blood group AB, both the antigens are present, no antibodies. Okay. It can receive from all the four types A, B, AB, and O and donate to only AB. Blood group O, no antigen, both the antibodies. Okay. It can receive from O only but donate to all. That is why universal donor. This is universal recipient. Understood? So if a person is having a blood group A, it means antigen A on the RBC membrane and the antibodies against B. So it cannot receive from B, okay, because against B it is having antibodies. It will receive from A and O. It will donate to A and AB. Okay. Blood group, because AB blood group, 50% RBCs are of A blood group and 50% RBCs are of B blood group. Okay. So a person having a blood group A, it can donate to AB because it is having 50% of RBCs having A blood group. Understood? Genotype of blood groups. I explained IAIO. IAIO means this is a recessive allele, and this is a dominant blood group is A. Okay. In a homozygous condition, it can be written as like this: blood group A. Okay. Blood group B, blood group B. Understood? In the previous slide, yeah, please remember this is O. O, O. Okay. Now, gene O is recessive gene which gives its expression in homozygous condition. It only gives in homozygous condition like IO, also written as. Understood? Blood group O is due to recessive gene. Blood group AB, codominance. RH factor. Another antigen is present on the RBC membrane, which is discovered by Landsteiner and Weiner in rhesus monkey. RH antigen is due to the dominant gene. Dominant gene, if it is present, then RH positive. If it is not present, then RH negative. Okay. So if one of the gamete possesses gene of RH factor, so its offspring will always be RH positive. Okay. In India, the percentage ratio of RH is 97% RH positive, 3% RH negative. In remaining world, 80% of RH positive and 20% of RH negative. Okay. So along with the antigens of A, B, AB, and O, RH antigen is also present on the RBC membrane. What is RH incompatibility? If a RH positive antibody is absent, okay, when RH positive antibody is absent for this antigen, okay, when in case of Rh positive person, antibody is absent. In case of Rh negative as well, antibody is absent. Okay, so antibody is absent in both the cases natively, mean in Rh positive as well as in Rh negative. But if the Rh positive blood is transfused to Rh negative, then the first blood transfusion complete successfully. But during first blood transfusion, Rh antibodies are formed in receiver's blood. So when Rh negative will develop this time an antibody which was not present before. Okay. So in the next blood transfusion, agglutination of blood takes place. Agglutination means clotting. Okay. If mother is Rh negative and father is Rh positive, then offspring will also be Rh positive. It is given that if one of the gamete is having Rh positive, then offspring will be definitely Rh positive. In this case, first pregnancy is completely successful because the umbilical cord is, after first pregnancy, umbilical cord is cut. Okay. But the umbilical cord is cut, it means there are placental pores. Because of these placental pores, there is a mismatch in fetal blood and the maternal blood. Fetal blood, maternal blood. So because of cutting of umbilical cord, pores are created over here, and the blood, it moves through these pores, which will mean the antibodies against, for example, if fetus is Rh positive, so the maternal blood Rh negative, it will create antibodies, and these antibodies will be transferred to fetal blood. Okay. So, but at first delivery, Rh antibodies formed in the mother's blood due to damaged blood vessel. So in the next pregnancy, death of fetus will occur in the earlier stage due to agglutination of blood, which is called erythroblastosis fetalis. This condition is known as what? Erythroblastosis fetalis. Okay. To destroy Rh antibody, anti-D vaccine is given. Anti-D vaccine. Okay. This anti-D vaccine basically it disrupt or it not let the maternal blood to make the antibodies. Okay. Prevent the formation of antibodies. Understood? So this is Rh incompatibility. If a Rh negative, when mother is Rh negative and father is Rh positive, then the baby will be Rh positive definitely. So if the Rh negative mother, it develops the antibodies, but because of first the umbilical cord was not cut before the first pregnancy, everything is intact, no mixed matching of fetal and maternal blood. But after cutting the umbilical cord, there are some, physiologic pores which are created between the fetal blood and the maternal blood, and that's why in the second pregnancy the fetus will abort. This condition is known as what? Erythroblastosis fetalis. Okay. To prevent this condition, we add anti-D vaccine or Rogum or Rogum or Rolin vaccines. These medicines are used. Rogum or Rolin or anti-D vaccines. Understood? So this is about Rh incompatibility. So we will be starting the next class, the next, I think, sorry, the next lecture on the muscular tissues. Okay. So till then, everyone, please take care, stay safe, Stay happy, stay at home. Bye bye. Take care.